Okay, so let's get started. And so welcome everyone today to the 2023 Sahyadri Outstanding Plan Postdoc Symposium. I am Ariadna Gonzalez Solis, an NSF PRFB postdoctoral fellow at the University of Wisconsin-Madison and the chair of Plan Postdocs. For those who don't know, we are a community of postdocs in plant-related fields with the goal to help each other to reach our next career stage. You can find more information about us in our website and social media. Everything is here on this slide. And on our YouTube channel, you will be able to find this symposium together with other videos from Plant Postdocs. And so today we're very excited to have the first Sahyadri Outstanding Plant Postdoc Symposium. This award is named after the Western Ghat mountain range in South India. Sahyadri refers to the benevolent mountains and is one of the biodiversity spots in the world. This award highlights exceptional postdocs in plant-related fields that are preparing to apply for academic faculty positions in the US this year. Along with an honorarium, each awardee will be paired up with a mentor that will help them through their job search. And so congratulations to our 4 awardees of this year. Jasprit will introduce them individually before their talk, but we just want to make sure to thank them and to wish you all the best in your job search. And thank you for sharing today your great science with us. And before getting started, I just want to also take the time to thank our sponsors of this award. So thank you to Sunil and Arif, co-founders of Plant Postdocs, and also thank you to Professor James Schnabel from the University of Nebraska-Lincoln, and also myself, who contributed to make possible this award for all the awardees that we have today. So with that, I'm just gonna stop sharing and let Jasprit continue moderating this symposium. Thank you, everyone.
Hello, everyone. So our first speaker for today is Hee-Kyung Aoun. Hee-Kyung Aoun is a plant molecular biologist, and her work is focused on how protein interact in plant cells. Currently, she is postdoc in the Sainsbury laboratory, and in future, she wants to start her independent research group. And her main question is going to be how immune receptors are maintained and assembled in plant cells. With that, I would like to welcome Hee-Kyung for her talk.
Thank you for the introduction, Jasprit, and I will share my screen. So first of all, I'd like to thank everyone. Thank you for the Plant Postdocs for the award, and I think it's a wonderful opportunity to receive this award, and also have the opportunity to have match up with a mentor, and also be able to present some of my current works that I've been doing. And I've never been to India before, so I would make sure that I will visit these mountain ranges when I have the chance to go. So the title of my talk today is Cooperation of 2 Immune Receptor Genes for Intracellular Pathogen Recognition. And as we all know, the plants live with other organisms, and they kind of interact with the organisms in multiple different ways. Some can be beneficial and some can be pathogenic. But today I'll be talking more about the pathogens that have deadly effects. So one of the most devastating example is the potato late blight that is caused by the filamentous pathogen mycetes that leads to these necrosions, necrotic lesions on the leaves and also on the tubers. And in order for these, although you do see plant diseases, if you look around you, you won't see that much of diseased plants, and that is because the plants are able to detect the diseases or the pathogens and protect themselves from it. And then the major question is, and how do these plants actually detect the non-self? How does it detect the pathogen and realize that they have to protect themselves and activate immune signaling? So many studies have shown that the recognition of non-self occurs in two different places. And the first is on the cell surface, and we call it the pattern-triggered immunity, which is derived from what are called the PAMs, or the pathogen-associated molecular patterns. This occurs mainly at the cell surface. On the other hand, there is the intracellular detection mechanism that is mediated by the nucleotide binding leucine-rich repeat proteins, or NLRs. And these NLRs are able to detect the proteins that enter into the plant cell, as depicted in here with the bacteria secreting a type 3 secretion system into it. And once we now know that the two tiers of the detection are mutually required for each other. So you do need both of them for full activation. But I'll talk to you more about the intracellular immune receptors today. So NLRs, what are the NLRs? NLRs are conserved in both plants and animals, and they have a distinct tripartite structure of a variable N-terminal domain and flanked by the N-bark domain, or the nucleotide binding domain, with the leucine-rich repeat. And that's why we call them the NLRs. One of the key features of the NLRs is that they are highly specific via the LRR domain, where the pathogen proteins bind to. And upon this specificity, there is also highly sensitive, because the pathogen proteins are very few that's entering the cell, and those can be detected by these proteins. So they're also very sensitive. And upon detection, what happens is that via the NB domain, or the nucleotide binding domain, there is immediate activation. And this immediate activation happens by oligomerization. The N-bark domain recruits or assembles other protomers of the NLRs into these forms called the resistosomes, and that is the signal, or the final assembly point of immune activation beginning. So most of these, as depicted in this diagram, they usually require one gene of several molecules of one gene. But what I'm studying is a slightly different. So in these cases, two NLRs are both required for signaling. So instead of having one protein, one gene, and multiple proteins of it that make into a resistosome, for these different types, we didn't quite know, and I've been contributed to understanding how these two NLRs work together. And so the first example is the sensor and helper NLRs. And the sensor NLR is called the sensor, of course, because the pathogen proteins bind to these proteins, whereas the helper NLR helps and executes the signal of the immune activation. And as an example of the sensor and helper NLRs, I've been studying the sensor NLR RPIMR3 and helper NLR NRC. So what we know is that these two genes are both required to confer resistance to late blight, or of the potato images that you've seen earlier. And my question was, does RPIMR3 and NRC both come together to oligomerize into resistosomes? So to study that, what I've been using was something called the Blue Native page, which resolves the protein complexes rather than resolving the protein molecules itself on the SDS page. And then this resolves the big oligomers compared to the smaller monomers. And if you follow this QR code, you will be able to read the protocol that we have recently published. So after trying this, what I first shown was that RPIMR3, which is the sensor NLR, is interacting with the pathogen protein called AWARDEES. And this shift in band that you see for RPIMR3 is also detected in AWARDEES3, but without RPMR3 co-expression, AVRMR3 does not make these higher order forms that you've seen before. So this suggested to us that what's happening is RPMR3 binds to AVRMR3. But what was important to understand was the fact that this did not correlate with let's say a pentamer or a hexamer of these proteins, suggesting that RPMR3 itself does not oligomerize. So next, we wanted to see from the helper side. So the helper NLR NRC was used, NRC2 was used, and we showed that upon activation of RPMR3 and AVRMR3, we do see a big shift in the oligomer of NRC2. And that suggested to us that NRC2 is forming the helper NLR resistosome, whereas RPMR3 does not. So this was a work in companion with Sophie and Kamoun group, and we have published these back-to-back in March this year. And our final conclusion, or our working model, is that sensor NLR binds to the pathogen proteins, which then activates the helper NLR, and then the helper NLR alone, without incorporating the sensor NLR, becomes an oligomer, and that induces disease resistance. But if you remember maybe earlier, that there is another type of two dual NLRs in working together, that's the paired NLR. Paired NLRs are different from the sensor and helper NLRs in that these are linked within the genome and they are often sharing a promoter as well. And in this case, the sensor NLRs often have an extra domain called the integrated domain that binds to pathogen proteins, whereas the executor NLR does not have those additional domains. So as a working model for the paired NLRs, I've been using the RS1 and RPS4 paired NLRs from Arabidopsis. And in these systems, RS1 functions as the sensor NLR with the additional worky transcription factor domain. And that this then is modified by pathogen proteins, AVRPS4 from Pseudomonas, and the POP2 acetyltransferase, which comes from Ralstonia solanaceae. And after this detection, the RPS4 then activates a signal. What we know from here, so I reran the Blue Native page, but using two different independent Arabidopsis transgenic lines that are tagged with RS1, RPS4, but you can see that flag tags are bound to both RS1 and RPS4 independently. And this enabled us to compare the actual migration patterns of both RPS4 and RS1 at the same time. And this is different from the previous sensor and helper NLR case in that even in the inactive state or without any pathogen proteins induced into it, the RS1 and RPS4 remain as an oligomer. And we further showed with sucrose gradient fractionation that regardless of the activation states, RS1 and RPS4 protein complex is about 600 kilodaltons, and that is expected to be about 4 molecules of RS1 and RPS4. So to put everything in a summary, I think in the plant immunity field, we have recently seen many discoveries regarding the resistosome formation of ZAR1 and ROC1. And these classic examples showed that upon activation, there is induced oligomerization using, and that is bound by the effectors. But my studies or our research from our group has shown that maybe there are other cases, for example, NRC2 may oligomerize without having the effectors bound, whereas the sensor NLR RPMR3 does bind to the effector but does not oligomerize. And yet we have another example in the case of paired NLRs in that the induced oligomerization doesn't really happen, and they are already poised to be activated. And so the take-home message today would be that not all of the NLRs work the same way, and we are trying to identify the more variable or the variations of activation of the NLRs. And with that, I'd like to thank everyone, especially Jonathan, for hosting me throughout the years of working in TSL, and also to the RS1 and RPS4 team, and also the RPMR3 and the NRC2 team within the group, as well as the collaborators, especially Sophie and Kamoon group, for sharing the genetic resources for the NRC2 work, and also to the funding bodies. Thank you very much. I think you are muted. Stop share. You are muted.
You're muted, Jaspreet.
Thank you. Yeah, if anyone have questions, please post it in the chat. Okay, so I'll ask one. So how do you want to take this forward into your own research, and what are the other components you think you will be bringing into your own independent research projects?
So I think what we're trying to understand now is how the NLRs are activated, and I've been thinking that maybe this could be a way to identify new resistance genes. So there are resistance genes. So there are about 150 of these NLR genes in Arabidopsis, and not all of the responding pathogen effectors are known in those 150. And if oligomerization or induced oligomerization is a key, is a mechanism, then we might be able to use that. And another way is of identifying the patterns or how the pathogen proteins behave in the cell in terms of these protein complex assemblies. If the pathogen proteins are binding to a certain host component, they would change or shift in their molecular weight, and that might be an indication of how it's acting in the plant cell. So those are some kind of ways that I'm trying to devise in terms of developing more more into my independent research.
Thank you so much. Thank you for the talk. Okay, so I think we can go ahead and proceed with the. All right, we have a question. So, do you intend to use cryo-electron microscopy in your analysis?
Yes, thank you for the question. Yes, that is our plan, and I think it is very difficult to purify NLRs in large amounts, and especially it was impossible to do so with E. coli cell purification. So with insect cells and other techniques are needed. But I think we have now come into a phase where we could purify sufficient amount of proteins of these NLR proteins from Nicotiana benthamiana. So we are quite optimistic that we will be able to put these into further cryo-EM analyses.
Thank you.
Thank you.
I'm just gonna wait for a second, just in case someone has a question. Okay. All right, we can proceed with the second talk, and then if there are some questions, we can always go over those at the end of the symposium. So our second speaker is Maryam Ishka. She's a postdoctoral researcher at Boyce Thompson Institute in United States, and her research work focuses on plant architecture and stress resilience. Maryam has also served as assistant features editor in the Plant Cell as an early career representative on Women in Plant Biology Committee of ESPB. So with that, we would like to welcome Maryam for her talk for the day.
Okay, thank you so much, Trevor. So hello, everyone. Good afternoon. I would like to first say thank you to the Plant Postdoc community for giving the opportunity to me to kind of share my research with all of you. So one of my projects that I'm working on it since I joined the Joukowsky lab is about hormonal modulation of tomato root system architecture under salt stress. So basically, during the salt stress, every aspects of plant growth and developments are impacted. As you can see, the impacts of the salt stress is basically clear. It basically impacts every aspects of growth and development. So the mechanisms involved, on the other hand, in the salt stress resiliency is quite diverse and multigenic. So in order to basically understand and investigate the mechanisms of the salt stress tolerance, so we need to focus on a specific, for example, tissue or organs that we hypothesize that it might contribute to the overall salt stress resiliency. So in our lab, so we are focusing on the root system architecture, and we are using that once at our discovery platform to basically investigate the plant's response to the salt stress tolerance. So for that, we basically leverage the natural variation within 230 tomato accessions of Solanum pimpelifolium. So these are the closest wild relative to the cultivated tomato. It's a reference genome sequence available. It has been sequenced by our neighboring lab at Boyce Thompson Institute, Dr. Faye Groups. And also the 230 accession has been genotyped at KAUST by a Mark Tester group. And the basically primary trial for field and control to several abiotic stresses shows basically indication of stress tolerance to a variety of stresses, including salt. So it's a good population to basically use for our investigation. So what we did, basically, we used a high dedication method, which we call it. It's very laborious method. So we analyzed the root system architecture of these 230 accessions based on the plate-based assay. So in brief, so we germinate the seeds on the our plates for 4 complete days, and then we transfer the seedlings to the treatment plates with and without 100 millimolar salt, and we start scanning the plates every day, and for a total 8 days. So after that, we have to use MHJ smart roots feature to basically trace all of the roots. And as you can see here, so to extract all of those root system architectural features. So as you can see, Magda started, I think, all of the tracing when the time was COVID. So, and there is nothing else to do. So it was a good time for root tracing. So, and then she basically moved to Boyce Thompson Institute and has her own lab. And then our technician, Haley Sussman, was take charge of, I think, all of those root tracing, basically. So when we were analyzing this root system architectural of this population, we could visually basically identify 4 different ideotypes of the root system architecture. However, when we use the traditional matrix of the root system architecture, such as lateral root number, main root length, lateral root angle, and etc., we couldn't basically to define these 4 visual ideotypes. So it seems that so there should be another way that we could quantify these root ideotypes, right? So for that, we used a mathematical model called Paratophone optimality solutions which quite often these models are used to basically describe underground networking system, the efficiency of, for example, the underground networking system in terms of wiring and the speed and the cost. For example, we have London Underground, which is fast but expensive. On the other hand, we have Glasgow Underground, which is slow but cheap. So there are some intermediates between those as well. So how we can basically calculate this Pareto front optimal solution for our root system architecture. So for doing that, so we collaborate with the mathematician, Dr. Arjun Chandrasekhar from University of Pittsburgh. Basically, so imagine that we have basically main root, which is highlighted in orange. And then we have lateral root meristem, which is shown by the blue dots here. So how we can connect those lateral root meristem to the basically main root base, right? So there are multiple ways of doing that. So one of them can be, the connection can be basically fast but expensive, right? It needs lots of building blocks and lots of root cells to be built up, right? Until they can carry the nutrients to the root base, basically, and then to the shoot. This is fast but expensive approach. So the example of the root ideotype for this can be broomstick, which we called it. On the other hand, the connection can be cheap, but it's slow. So you can build those building blocks along the way as you go on, right? But it takes a lot, right? Time to kind of to transport all of those nutrients from different lateral roots to the root base and then to the shoot, basically. And there is always an intermediate solution as well. So interestingly, when we use Pareto front to basically describe root system architecture of these 230 extensions, we found very interesting results. So first, I would like to basically explain the Pareto front model. So basically, in the x-axis, we have wiring cost, which is the cost for, let's say, building block or the number of cells that require to be built, right, in order to transfer the nutrients. And then in the y-axis, we have conduction delay. So how fast nutrient can be transported, for example. And then we have the dashed line, which is the Pareto front solution line. And the Pareto front has two components on it. Alpha, which is those dots on the Pareto front line. So the alpha is basically the location of each individual on the Pareto front line, right? So it ranges between 0 to 1. For example, alpha 1, the example of alpha 1 root ideotype is the cheap strategy, right? The telephone pole. So it doesn't need very, it doesn't cost much, but also it's very, for example, slow, right? Or the alpha 0, it's the root ideotype that is much more expensive. You need to build more cell types, but it's fast. The transfer can be very fast. And there is another component of the this Pareto front model is the distance, those red X. So basically the distance of each individual to the Pareto front. The closer the individual is to the Pareto front line or to the dashed line, you are more optimal to being efficient, basically, under some circumstances. So when we used Pareto front location component to basically analyze or explain our root system architecture efficiency, we saw, oh, okay, the results are very noisy, right? So, for example, a stress started with a very cheap strategy, but then toward the end of the stress, as you can see in the salt stress, so they're choosing much more expensive strategy, right, to invest. However, when we did basically use Pareto front distance for root system architecture, we saw very nice pattern. So in brief, basically salt stress makes root system architecture to be optimal, to get closer to the Pareto front optimality solution, which was very interesting and also makes sense, right? Under the salt and under the stress, plants are limited in their resources, right? They can basically minimize their growth and balance and think kind of smartly how to utilize the nutrients and to distribute it growth, right? So now the question is, what's the relationship with this Pareto front optimality and salt stress resiliency, right? So for that, we are doing right now GWAS on Pareto front basically component. And so another things that today my talk is specifically focused on that ones, can we modify Pareto front location? Or in simple words, can we change one ideotype to the other ideotypes, right? For example, change stress resiliency by hormonal manipulation. So, and is there any optimal root system architecture under the salt stress? So we use sodium accumulation as a proxy to basically investigate that questions as well. So for that, I chose 3 accessions, 1 cultivated tomato and 2 Solanum pimpelifolium, which we knew from our root system architectural screen. One of them is wild sensitive, means it's sensitive to the salt stress, and the other one is wild tolerant, which is salt tolerant. And basically we use the same strategy. We grow them, germinated them in the plates, and then transfer them to the treatment plates, and then scan every day, and then extracted those root system architectural features. So when we analyzed basically those root system architectural features, we found very interesting results regarding lateral root features. While I think the cultivated tomato showed a salt-tolerant index increase for the lateral root number, our wild tolerant showed increase in salt-tolerant index for lateral root lengths. That means these 2 salt-tolerant accessions utilizing 2 different strategies, while one of them increasing the number of lateral root, the other ones basically elongating the lateral root lengths under the salt stress. So that was very interesting. So I generated basically, in order to understand what's the genetic components of this variation. So I did a reciprocal process and I created the F2 population, and I screened the F2 population of these 2 tolerant accessions for root system architectural traits, and I could identify 4 ideotypes, which alongside with their parents, I sent for the bulk segregant analysis. And at the meantime, I also did RNA-seq on these 2 tomato accessions, which are salt tolerant, but utilizing 2 different strategies. So to kind of understand, so what are the commonality between these bulk segregant analysis and then RNA-seq experiments. So as I mentioned, so another aspect of my research was hormonal manipulation. Can I change one ideotype to the other ideotype? So I started with a screening root system architecture of these 3 accessions with all of the hormones. So among them, I found that basically auxin treatment alters root ideotypes, right? So it basically changes the root to whatever root ideotype the accessions has. It changes all of them unanimously to basically a telephone pole, which is marked by producing lots of lateral roots, but at the same time very short in all of the accessions. More interestingly, when I analyzed basically the sodium accumulation in collaboration with Miguel Pineros at Cornell USCI, so we found that IA treatment basically almost increased sodium accumulation in all of the accessions in both root and shoot tissue. So it means that IA treatment is not good for the salt stress, right? So whether auxin creates more entry gates, right, that can trigger sodium into the plants, and what are the anatomical configuration in the root, and does auxin triggers, I don't know, suberin degradation, and what are the gene regulatory networks? So we are still trying to figure that out. There are some groups, Dr. Nico Gelender, that are basically, they show that there are some links between auxin and salt stress. Basically, auxin helps both suberin biosynthesis and also degradation, which is very interesting. So we would like to see also that results in our hands as well. So among the other hormones that I tried, I found that ethylene basically has the contrasting impacts on the salt stress. Basically, the response to ethylene treatment varies significantly across accessions. So if we just look at the lateral root number under the salt stress, we see that different accessions had different response. So one of them, for example, if you look at the cultivated tomato, in response to the low amounts of ACC, we see decrease in lateral root number. However, for example, if you look at the sensitive ones, so in response to the ACC, the lateral root number increased. So there are variations here. And more interestingly, when I analyzed basically the sodium accumulation due to ethylene treatment, so we saw that unanimously, basically ethylene mitigates sodium accumulation. So that means it's good for plants, right? Well, under the salt. So again, what are the anatomical configuration? Why that changes happens? Do we see any increase in suberization, and that's why the sodium ion cannot get into? And what are the gene regulatory networks we hope to get back from our RNA-seq results, basically? So basically, we showed that there is a diverse response in salt stress tolerance, while IAA basically increases sodium accumulation and it's not good for the salt stress response. ACC has the contrasting impacts, right? So in the future, right now, basically, I am investigating the effect of these two hormones specifically on tomato shoot growth under the saline soil, because that was the plate-based assay, right? I would like to see what's the long-term impacts of these two hormones on the overall tomato shoot growth, right? And basically, I would like to overlay my RNA-seq and bulk segregant analysis to identify potential genes that are involved in the lateral root development under the salt stress and start basically functional characterization for those genes as well. So at the end, I would like to say thank you to my lab members, especially to undergraduates that are taking charge of my root tracing, which I really appreciate that, Trent and Stacy, and our collaborators in these projects. And thank you, all of you. Last but not least, so I would like to invite all of you, please register for Gordon Research Conference in Salt and Water Stress Implants, which is coming up next year, August, in May. So Trevor, which is here and presenting, and me are chairing the Gordon Research Seminar of this conference. And please reach out to us if you have any question. That's a great scientific environment. Come and try to share your knowledge and receive, more importantly, great feedbacks from the great scientists. Thank you so much. And I'm happy to take any questions.
Thank you, Maryam. All right, so we'll just get to the questions. So I think one question we have is how many accessions show, I think it's parietal, forgive me if I'm pronouncing it wrong, front optimal under salt stress in the population.
So how many accessions? So okay, so I described it. How many accessions showed parietal front optimality?
Optimization under salt stress.
Yes. So basically, we used those parameters, alpha and distance of the parietal front optimality solution, to feed into the GWAS. So the basically results that I showed here it's for all of 230 accessions, right? These are all for 230 accessions that unanimously showing, I think, the same pattern. For example, the bottom graph, which is a distance component, show very nicely the salt stress, which is marked by the red. So almost all of the accessions, right? They are basically getting closer to the optimality, right? It means that salt stress basically forces plant to be optimal. There is no other way, right? They cannot grow more, right? You have to minimize, you have to think wisely to survive, right? And I think kind of makes sense. So we can go back to those data and identify those individuals, right? So which accession, for example, have this alpha or that alpha, but these graphs are for all of the accessions that we performed.
Okay, so next question we have is why you have grown plants in 1/4 MS medium? Is there a specific reason? And another question is, besides sodium, do you find any other macro or micro element which are increased during oxygen treatment?
Thank you so much. These are really great questions and technical questions. I love technical questions. So when I joined the lab, so they grew the tomato in 1/4 MES because they say, oh, okay, tomato is so strong, right? Compared to Arabidopsis, which we use usually half a MES, right? To grow and germinate the Arabidopsis. So I just adopted that methods and it works. So also we use less MES, right? Yeah, I think there is no special reason. So it's just, it was there and I used it. And I think there is not much difference. Sometimes if I am in rush, wants to just germinate it because germination in separate plate, right? And then you have to transfer it. So I also can use germinate those tomato seedling in half a MES like the Arabidopsis as well, right? So there is no difference basically. But if you kind of cut down the MES, so it gets less salty, which is nice. So and did I check other elements basically besides So basically what I do from my ICP-MS, I look at sodium, potassium, and the sodium-potassium ratio, which is important for the salt stress. I haven't looked at other micronutrients, but I love to look at them because there is a relation between sodium and calcium. There is a relation what happens to the chloride, but you know, they are very, you know, interestingly, my previous postdoc was on micronutrient deficiency, and it can go anyway, right? So you don't know how to interpret the results. One of them is increased, the other one is decreased. So I just try to be simple and focus on sodium, potassium, and the ratio just for this set of, I think, experiments. But definitely it's a nice things to kind of look more. I think we cannot hear you, Jasper, but I'm assuming that I can stop sharing.
Sorry. Right, so we'll move on to our next speaker. Our next speaker is Nathalie Aoun. Nathalie is a postdoctoral scholar at University of California, Davis, and her current research investigates infection and colonization mechanism in plant pathogenic Ralstonia. So with that, would like to welcome Nathalie for her talk today.
One second, I struggled for a minute to share properly. All right, can you see my screen and hear me well?
Yes.
Awesome. All right, so hi everyone. Thank you again for the Plant Postdocs for this great opportunity to be selected for an award and also to present my work today. So for today, I decided to present one of my recent research projects. So you'll be seeing fresh results from my work. And the talk will be focusing on investigating the adaptive mechanisms underlying plant colonization by Ralstonia. So Ralstonia are soil bacteria that infect tomato plants or any, like a large number of plants. And you can see here on the left side, a tomato plant that is infected with Ralstonia showing wilting symptoms and dying. So Ralstonia belong to a species complex that are known to cause severe crop production losses that could threaten global food security. For example, in 2005, Ralstonia had infected 1.5 million hectares of potato fields and has caused estimated losses of around $1 billion of potato production. And here on the right side, you can see a potato that is infected with Ralstonia, showing the bacterial goop as the white stuff showing here. So Ralstonia have a high pathogen diversity and they have a wide geographic distribution. They like warm and temperate climate conditions. They are divided into 4 different groups called phylotypes, and these groups are based on their geographical origins. So as you can see here on this worldwide map, we have phylotype 1, 2, 3, and 4. So as I mentioned before, Ralstonia have a wide host range as they can infect 400 different plant species, and they can survive in soil and water, which makes them very hard to control. And so until today, there are no disease control strategies against these bacteria. So once in the soil, Ralstonia can sense the host through the roots and then colonize the roots through any wounds or injuries, and then colonize the xylem vessels where they can secrete exopolysaccharides and clog the vessels, which will alter the water transport and lead to the wilting and the death of the plant. And here on the right side, I'm showing you a few examples of wilting symptoms on different plants caused by Ralstonia. So what the low power lab focuses on in general is to study the microbial ecology of Ralstonia and to understand how these bacteria survive and adapt to their host. And in my postdoc project, particularly interested in investigating the Ralstonia adaptive mechanisms to the tomato host. So I'm interested in studying if Ralstonia use similar mechanisms or host-specific mechanisms to infect plants with different levels of quantitative disease resistant response. And to study this, I focus on two main approaches, which are the high throughput forward genetic screening and the microbial genome-wide association mapping, or GWAS. And for the sake of time today, I will only focus on the first approach. So when I first started working on this project, I needed to define a panel of different tomato varieties that I will be focusing on. And so our collaborator, Robin Roberts from Colorado State University, has previously screened different tomatoes with different phytobacteria, such as Clavibacter, Pseudomonas, and Xanthomonas. And there was a group of varieties that have shown resistance or tolerance to these bacteria. So once I saw these results, I was interested in testing these tomatoes with Ralstonia. And so these tomato varieties are listed here in the legend. And I also added to this list the Moneymaker and Hawaii 87996 tomato varieties, which are extensively studied in the field. So what I did is that I screened these tomato varieties with two representative strains of phenotype 1 and phenotype 2 of the Ralstonia species complex, which are the GMI 1000 and the IVS-BF 1503 strains. So I inoculated these tomatoes at the soil level, and I screened them for wilting symptoms over 2 weeks. So on the x-axis here, you have the days after inoculation, and on the y-axis, you have the disease index scale that goes from 0 for totally healthy plants and 4 for totally wilted plants. And so this scale is defined based on the number of wilted leaves over the total number of leaves in the plant. So the results show that with the GMI 1000, we have a more diversity of tomato response to the bacterial aggressiveness, unlike with the IVS-BF 1503, which is more aggressive on the tomatoes. And so looking at this, this reinforced my questions about if, for example, the GMI 1000 strain used different strategies to infect the tomatoes with different levels of immune pressure, and if the GMI 1000 and IVS-BF 1503, so strains from different phenotypes, use common mechanisms or different mechanisms to infect a host. So to answer these questions, I use a high throughput for a genetic screening approach called the random barcoded transposon mutant sequencing, or RBTN-seq, which is based on using a library of transposon mutants. So in this library, we have bacterial mutants for thousands of genes, covering around 80% of the bacterial genome. And each bacterial mutant has a mutation in a specific gene and a barcode that is unique based on 21 base pair. So having a barcode for each mutant allows us to track these mutants in any condition. And so in the lab, we have these libraries in different strains background, particularly in the GMI 1000 and IVS-BF 1503. So what we basically do is that we revive these libraries in rich media, and we save some of it as a time-zero control, and then we use the rest of it to inoculate tomato plants. And once wilting symptoms start appearing, we harvest the stems, we homogenize them, we do this detox juice, and then we do several filtration steps to harvest bacteria back. And then we extract the DNA from the time-zero control and the bacteria harvested from the plants, and we send them for Illumina barcode sequencing. So based on the Illumina barcode sequencing, we can calculate a gene fitness score, which is the log2 ratio of the mutant abundance in the condition over the mutant abundance in the time-zero control. And this is an example of the results we get. If a fitness score is a positive, it means that the mutant had a gain of fitness, which means that the corresponding gene is hindering the bacterial growth. But if the fitness score is negative, it means that the mutants had a loss of fitness and the corresponding gene is required for bacterial growth. Now, if the fitness score is around 0, it's a neutral mutation. We don't really focus on these genes. So I conducted RBTN-seq experiments in the GMI 1000 RBTN-seq mutant library background and the IVS-BF 1503. But here I'll be only showing results from the GMI 1000 experiments. And what I noticed is that there's a set of genes that seems to be commonly contributing to bacterial growth inside the plant during infection. And these are membrane transporters. And what's interesting is that these membrane transporters have a paradoxical contribution, meaning that some of them had a positive fitness score and some of them had a negative score, and they were still contributing to fitness. So these membrane transporters belong to 3 distinct families, which are the ATP binding cassette transporters, the resistant modulation division, and the major facilitated superfamily. For instance, in the ABC category of transporters, these 2 mutants had a gain of fitness, meaning that these transporters are hindering the bacterial growth, while the other one are required for bacterial growth. And it's kind of interesting because we would expect them to have a similar function during the host infection. But what we know so far is that these transporters are known to be energy dependent, and they are known to expel or import, they can import nutrients or expel toxic compounds. And they were mainly studied in human, animal pathogens, and they were associated with multi-drug resistance. But in some cases also in phytopathogens like botrytis and other pathogens, they were associated with virulence. But until now, we don't really know how they function in Ralstonia. There's one example of 2 genes encoding for RND and another type of transporters that have been shown to contribute to the bacterial virulence, to Ralstonia virulence, but we still don't know why and how. So what I'm currently trying to test is to see if the transporters that promote fitness are being more efficient in expelling toxic compounds secreted by plant defenses, or maybe they have a higher efficiency of importing more nutrients for cell homeostasis. And for the transporters that are challenging the bacterial growth, we're currently trying to see if they are being targeted by plant defenses, or they are failing to expel toxic compounds, or maybe they're failing in fulfilling their other basic functions. Besides looking at the functional role of these transporters, I'm also interested in studying the evolutionary processes of these transporters. I don't think I have enough time to explain this, but basically checking the presence of homologs of these transporters in different Ralstonia strains and neighboring bacteria to see, to understand better their organization and biological role. And to conclude, I want to say that RB10-seq is a powerful tool to study bacterial gene function in any condition, and that transporters seems to be a common mechanism that contribute to Ralstonia growth during infection. And we're still trying to validate their functions, and that understanding their evolutionary processes and checking their genetic neighborhoods could bring more insight into their function. So stay tuned for any updated results from this project. At the end, I'd like to thank the Low Power Lab, especially my PI, who's always a very supportive PI, and also all the undergrads and master's students who've been working with me on creating knockouts of these transporters. This summer, we had our own cloning bootcamp, which was really fun to help them learn how to do cloning. And I'd like also to thank our collaborators and the Plant Postdocs community for this award and for this chance to present today. And thank you all for your attention.
Thank you, Nathalie. If anyone has questions for Nathalie, please go ahead and post it in the chat box. Okay, so we have a question that during initial days of infection in tomato with phylotype 1 and 2, you get same difference in varieties, but on later stage phylotype 2 seems to become same in all varieties, while phylotype 1 is showing difference. Have you quantified any metabolite in later stages of tomato?
Thank you for the question. This is very interesting. We don't, the purpose of this screening was just to check if these tomatoes had different levels of defenses against the bacteria. And the goal was not to check deeper which metabolites are contributing to these differences.
Okay, so another question we have is, did the transporters affect mutants' fitness? Sorry, Did the transporter mutant affect fitness of the bacteria itself by any chance?
Yeah, so thank you for the question. By fitness, what we check is the actual growth. So how much these mutants were able to grow in the condition. And what we're currently trying to do is to validate the phenotype we saw with the RBT and seq experiment by individually testing those mutants. Because in the RBT and seq experiments, we are testing all the mutants at the same time. So like if there are some redundant functions, we will not be able to see that when we do the RBT and seq experiments. So we're trying to validate these individual functions by creating individual mutants of these transporters in Planta. And this is actually ongoing right now.
So we have limited time, so we'll go ahead and move on to last talk of the day. And thank you, Nathalie. Okay, so our last speaker for today is Trevor Nolan. Trevor is from Duke University. He's a postdoc, NSF PGRP postdoc in Philip Memphis lab. His work is on using time series single cell RNA sequencing to unravel the interplay between cell identity and hormonal signaling in Arabidopsis roots. With that, we welcome Trevor for his talk today.
All right. Thank you so much for having me. I'm really excited to be here with you all, and I thank the Plant Postdocs for organizing this and putting together these awards. So today I'm going to share with you some of my postdoc work, which really sits at the intersection between hormone signaling and developmental biology. And broadly, I'm interested in how cells in multicellular organisms collectively coordinate so that they can execute their developmental programs amidst the complex challenges of dynamic environments. So in this process, cells have to pass through different states as they differentiate and they acquire identities. But how do these cell identities, cell states, and signals like hormone responses affect one another? To study these processes in the context of a growing multicellular organ, I use the Arabidopsis root, which is shown here, as a model system that's easily accessible for developmental studies. So one of the nicest things about these roots, if we zoom in, we can see in the growing root tip, this kind of beautiful microscopy, where cell proliferation occurs through divisions at the tip of the root in the meristem. And then cells dramatically enlarge in the elongation zone. And together, these processes of cell proliferation and cell expansion determine the extent to which a root grows. So this means that each of the different cell types, which are organized as these radially as cylinders, form a developmental timeline along the longitudinal axis of the root, from the youngest cells in the stem cells to the oldest cells in the maturation zone towards the shoot. But until recently, it was difficult to understand how gene expression changes across both these cell types and developmental stages. So in the past few years, I've been using single cell RNA sequencing to examine gene expression in individual cells. And if you aren't familiar with these techniques, this is essentially putting a cell and a barcode together along with chemical reagents that allows us to access the transcriptome of thousands of individual plant cells to generate a gene by cell matrix, which we can then use to begin to reconstruct developmental progressions, for example, of an entire organ like a root. And this is exactly what we did in a collaborative effort led by Rachel Shahan and Chelwei Xu, where we integrated more than 100,000 single cell transcriptomic profiles of the Arabidopsis root. Our single cell atlas captures the major cell types of the root, for example, the vascular tissues in the center of the root, the endodermis and the cortex that share a common stem cell and then form these two cell types, the trichoblast and atrichoblast in the epidermis, and the columella and lateral root cap. What's more, we can also trace the developmental progression of these cells. So the youngest cells, the stem cells, are found in the tip and the base here, in the meristem, followed by the elongation and maturation zones, as well as the columella and the lateral root cap. So by having these data, we've been able to delve into the continuous progression that seems to be involved in development, as well as gain insight into things like mutants, where we can identify new aspects of cell identity acquisition. What's more, we can use this Arabidopsis root atlas as a reference for any new experiment that we perform in the root. So similar to a reference genome where we can align reads, we can actually align cells to this reference transcriptome and identify their cell type as well as their developmental stage. So when I came to the lab, I was really interested in performing time course single cell experiments. And as a first test case, I chose Brassinosteroids, which are plant steroid hormones that are important for almost every aspect of plant life, including root growth and development, where they promote cell elongation or cell expansion. The Brassinosteroid signaling pathway has been relatively well studied, and we know that it involves binding of the hormone to plasma membrane receptors that initiate a series of signal transduction events that eventually activate downstream transcription factors, BES1 and BZAR1 in the nucleus, to regulate thousands of genes. But what we didn't know was how this BR regulatory network is wired in different cell types or developmental stages to carry out particular responses. So to investigate this, I set up a sensitized system where I first blocked endogenous brassinosteroid biosynthesis with a chemical called BRZ or brasinozole. And then that allowed me to add back brassinosteroids at particular time points. And you can see here that when I treat with BL, brasinolide, the most active brassinosteroid, that we can see rapid accumulation of the downstream transcription factors in the nucleus and a corresponding acceleration in root growth and cell elongation. Using this system, I then performed a time series of brassinosteroid response using single cell RNA-seq. So I first blocked this endogenous biosynthesis, and then I profiled the response on different time points ranging from a half an hour to 8 hours. And as you can see here, we could then examine what happens across different cell types as well as developmental stages. And by doing so, we saw that more than 8,000 genes respond to brassinosteroids, many of which are specific to certain cell types and or developmental stages. So for example, the epidermis, which was well known to respond to this hormone, seemed to be affected across developmental stages. But the next layer in, the cortex, seemed to be responding at a particular developmental phase, specifically the elongation zone. And we saw there that cell wall related genes were upregulated by brassinosteroids. For example, if we look at a reporter, CBIP2, that's induced by brassinosteroids in the elongating cortex, we can see that this comes on just as cells are beginning to expand or elongate, suggesting that this could be important in controlling root growth. To look more broadly at these, we examined a signature of 107 brassinosteroid-induced cell wall-related genes in the cortex. And here I'm plotting the cell wall gene expression score, where a higher score indicates increased expression of this set of genes. And you can appreciate here that we have a set of highly responsive cortex cells with higher expression of this set of genes relative to cells in other cell types. And we then asked, how do brassinosteroids affect these cells, and how do they relate to the developmental progression of cells along the longitudinal axis of the root? When we did this, we saw that along our time course, there was an increase in the percentage of responsive cells, and that this coincided with a shift to the elongation annotation of developmental zone, suggesting that the turning on of these genes is associated with increased cell expansion or elongation. Well, how can we test this? So in order to do that, I worked with Nemanja Vukasinovic and Jenny Roshanova in Ghent, where we used tissue-specific CRISPR of the brassinosteroid receptor BRY1 to test this function in brassinosteroid signaling. You can see here, essentially, we can erase BRY1 in specific cell layers and then ask what happens. And when we did this in the cortex, we found that the merostems, the tip of the roots, were relatively unaffected. However, the final cells were much shorter, suggesting that indeed brassinosteroid signaling is important in this particular cell type at a certain developmental stage where the cells are going through expansion. Well, how does that work? What are the gene expression changes underlying this? In order to infer these, we set out to look at brassinosteroid gene regulatory networks under specific cell types, developmental stages, and time points using our single cell RNA-seq data. And this allowed us to really zoom in on this important time point. The 2 hours of this response peaked in the elongating cortex and identify 2 families of transcription factors that were controlling these cell wall related genes. Among these was HAT7, which was the top ranked transcription factor that I'll focus on today. And when we looked closer at HAT7, we found that it was enriched in the elongating cortex, and particularly turned on right as these cells were beginning to expand or elongate. And when we knocked out HAT7 along with 3 closely related genes, we then saw that the plants had reduced cortex cell size, suggesting that HAT7 promotes cortex cell elongation. On the other hand, when we looked at the morphology of the roots, surprisingly, even though they had shorter cells, we saw that the roots were about the same length as wild type. Well, how can that happen? Root growth is a product of both the cell size and the number of cells that are produced through divisions. So we hypothesized that maybe there was an increase in proliferation that compensates for the reduced cell length in these quadruple mutants. And indeed, when we examined the meristem sizes and the meristem cell numbers, we saw that HAT7 quadruple mutants had significantly more cells in their meristems compared to wild type. And this brought on a whole series of new questions that I've begun to think about. The first of which is, how do cells actually switch from proliferating to elongating? Are there universal versus cell type specific mechanisms similar to what we see in HAT7? And moreover, how do cells communicate to coordinate the growth between the different cell layers? So what are the cell-cell signaling mechanisms involved in these? Based on these questions, I'm thinking about 3 different areas moving forward. So the first area is to try to understand how this spatial temporal shift operates within the 3D context of the root. So shown here is a beautiful image from Tatsuya Nobori at the Salk Institute, with whom I collaborate with, where we're using phytomap spatial transcriptomics to profile gene expression in individual cells within intact 3D tissues. And I'm interested in using this kind of technology to connect gene expression to morphological changes and to understand regulators like HAT7 that I've discovered. In addition, I'm interested in looking at the coordination of these cell state transitions between neighboring tissues. So for example, we saw when we knock out Brassinosteroid signaling in the epidermis that the neighboring tissue, the cortex, has these dramatic changes in cellular morphology. But how are those sensed by a neighboring tissue? So how does that tissue know that the neighbor has changed? How does it respond to it? And how is this important for coordinated organ growth? And finally, I'm using large-scale CRISPR screening to discover factors involved in controlling root growth and understand their role in cell division and cell elongation, with the ultimate goal of being able to understand these genetic networks to a point that we can engineer roots to grow longer or shorter to suit different environments, for example, using synthetic gene circuits. So in summary, today I told you about how we created a single cell reference of the Arabidopsis root that allows us to examine gene expression patterns across both cell types and developmental stages. And furthermore, by adding a time series component, we can reveal spatial temporal regulators of root growth like HAT7. And lastly, I'm really excited looking forward about the combination of single cell spatial approaches and live imaging to watch development happening in real time. And I think this is going to allow us to really delve deep into these mechanisms controlling the switch from proliferation to differentiation and how cells are communicating as organs grow. So I want to dedicate my talk today in this work to my postdoc mentor, Philip Bemphy, who passed away in September of this year. So Philip was an exceptional scientist, an amazing mentor, just an all-around incredible person. And I'm so glad to have worked with him these last few years. And I also want to thank all the many people in our lab, our collaborators, and our funding sources. And I want to mention, if you're interested in our data, you can view our interactive browser. You can also access our code and tutorials at the following links. And with that, I will be happy to take any questions that you might have.
Thank you for great talk, Trevor. I must say, like those, the single cell maps, it looks like, you're looking at the space, the pictures from the space. It was very interesting.
Thank you.
Yeah. If someone has questions, please go ahead and post it in the chat box. In the meantime, I'll go ahead and ask one question which I have. So what was your strategy for doing the site or tissue specific mutagenesis in Arabidopsis, and then for your bigger screening of mutants, are you thinking about like similar strategy or like targeting multiple tissue at a time?
Yeah, so these are kind of two different flavors of gene editing. So the tissue-specific CRISPR expresses Cas9 under different promoters. So this was actually using the CRISPR TSKO system developed by Tom Jacobs and Moritz Norwak at VIB Plant Systems Biology in Ghent. And so you have Cas9 under a particular promoter. It's only expressed in that cell type, and that means that you're only making your mutations in the particular cell type of interest. Mm-hmm. On the other hand, the large-scale screening that we're using is actually, it's called a multi-knock approach. It's developed by Aoun Shani. And there we have 6,000 guide RNAs targeting 2,000 transcription factors. So these are all driven by one particular promoter more broadly, and we're generating stable mutations. Mm-hmm. But the fun thing is that you generate, so we target more than one gene at a time, and you have a random combination or random guide RNA in each plant, which means that you can find these really interesting phenotypes, and then you can quickly correlate them back to which genes you're targeting and understand what mutations might be causing those phenotypes.
Sounds good. All right, with that, we would like to thank, okay, so we got one more question. We're just gonna take it. So we have a question that, very nice talk. I always have this question in my mind concerning to how this technique can precisely differentiate the transcript expressed in one cell versus in another.
Okay, so I think the question is a technical one about how we can tell that a transcript is expressed in a particular cell type and not another. And I think this is fairly worked out in single cell genomics at this point. So actually inside each of those individual drop codes droplets is a unique barcode that when we do the library preps, we can match the transcripts back to which cell it belongs to. So assuming there was only one cell in each droplet, then we can be pretty sure that the transcript came from that cell. The trickier part is that we actually have to use the transcriptome, which is a fairly sparse measurement, to identify which cell it is to begin with. And for that, we've really leaned on things like fluorescence activated cell sorting that was done by people like Ken Birnbaum and Siobhan Brady in our lab, as well as dissection of roots, to have a sort of ground truth of what different cell types or developmental stages look like. So without that infrastructure, it becomes much harder to know which cell is which.
And I actually, like, a related question came into my mind is like, so you looked at the time series, right? And then, you know, you might have seen like some of the genes which are expressed in particular cell type, like over the development. Is it possible that at certain developmental stage, they are just expressed in one type of cells, but over the time they're expressed in like different cell types as well? Like, especially like metabolic genes or something. Just curious.
Yeah, I think that's certainly possible. In terms of the Brassinosteroid time course, we didn't see a ton of that. We saw that the cell identities and developmental stages were modulated, but were somewhat stable. But in other time courses that we've done, for example, we've been doing a time course where we sample every 12 hours of roots grown from 3 days to 10 days. And there we see that there are some genes that start out being very specific, that then become more broad or that change their expression patterns in unexpected ways. So I think that can be kind of cool to think about something like how does a root age and how do those changes in gene expression relate to the morphology and the biological processes happening inside those roots.
Yeah. All right, with that, let's thank our speakers and congratulations to the awardees for this award. And then we wish you good luck in, you know, your future search. And then I hope you have like amazing time with your mentorship project. And let's thank you everyone who joined our seminar today, and stay tuned for our future seminars and events. Thank you.